Welcome back to part 3 of our first lab set of videos. Now we're actually going to jump into the actual histology portion of today. We're going to look at several different types of epithelial tissue. We're going to point out some structures. We're going to talk about how those structures allow this tissue type to do the functions it's associated with, and we'll talk about where in the body these different tissue types are. So you'll notice, again, remember from that second video, we talked about how we classify epithelial tissues. We have two major terms that each epithelial tissue gets. We have the shape of the cell, and we have the number of layers. So all of our epithelial tissues will be some combination of simple, stratified, or pseudostratified, and columnar, cuboidal, or squamous. Putting those two terms together, we are going to get a description of what the actual epithelial tissue looks like based on the cell shape and the depth of the tissue. So looking here, we are going to have our simple squamous epithelium. That is our one-layered, funky-shaped, flat cell epithelium, simple cuboidal, simple columnar. Then we have that pseudostratified. Remember, this is going to be the type of epithelium where all the cells touch the basement membrane. Not all of these cells are going to touch the lumen, though. Then we have our stratified squamous epithelium, and these come in two types. We'll talk about that more when we get there. Stratified cuboidal, and then because this is biology, we always have to have one guy who does his own thing. So we have one type of epithelium that actually doesn't get named in that nice dichotomous two-option naming system. We have our transitional epithelium, which we'll end on. Jumping into this first guy here, let me get myself out of the way a little bit. We have our first epithelial tissue type. We have our simple squamous epithelium. Now, the word squamous actually comes from the root squam, which means scale. So think of squamous cells as looking like scales, like the scales you'd see on a snake, these flat structures that take funky shapes. So our simple squamous, simple one layer thick, squamous flat cells, they're going to bulge up at the nucleus. Again, think of like a fried egg where you have the white, which is very flat, and then the yolk is a little bit taller there in that fried egg. Same idea here for the squamous cells. We're going to have a very flat area and then just a bit of a bulge there where the nucleus is. Now, if we think about a cell that's really flat and can take any sort of shape, this is actually a really useful cell for things like rapid diffusion or transportation of substances. So if we think about a really flat cell and we want to say get an oxygen molecule across it, well, it's going to be much quicker to get an oxygen molecule to diffuse through a squamous cell, which is flat, than something like a cuboidal cell or a columnar cell where there's more internal volume that oxygen molecule would have to traverse across. So our simple squamous epithelium are going to be found in places that we want really quick diffusion or transport of substances through the membrane. It's also going to secrete lubricating fluids such as serous fluid. So we're actually also going to find these simple squamous epithelium lining things like our blood vessels because it's going to create a very slick surface for blood to move past. It reduces friction, it reduces drag of blood, and makes blood flow more efficient through those vessels. So thinking about places we want rapid diffusion of substances, places we want to reduce drag, well, we're going to find simple squamous epithelium in places like the alveoli of our lungs where we're doing gas exchange, also in places like our kidneys where we want the really quick filtration and reabsorption of things like water, salts, toxins, to create our urine. We're also going to find this in places like the inner lining of our heart and blood vessels, where again, we want that blood to be able to pump through that tissue type, through that lumen without a lot of drag. And we'll also find this tissue in places of our gastrointestinal tract. So looking at this microscope image of the lung tissue, here we can see it drawn out. What we can see here are all of these areas here, these funky kind of borders, those are all going to be our simple squamous epithelial tissue. And you'll notice it's a continuous layer of tissue, a continuous sheet of tissue, one cell layer thick, and these cells can take any sort of shape in that tissue layer. They're all going to be pretty flat, pretty smushed, and just kind of smush in how they fit. These are each going to be our squamous epithelial cells. So when we're talking about the actual cell in an epithelial tissue, that cell is going to be named after its shape. So our cells in simple squamous epithelium are going to be squamous epithelial cells. Now, these areas here, the lumen, where the light passes through, those are going to be the alveolar space, where we're actually having gas, atmospheric gas, go into the lungs, and we're going to be able to have really rapid diffusion of oxygen into our squamous epithelial cells, and pretty rapid diffusion of, here we see a blood vessel, carbon dioxide out of our blood vessels through our simple squamous epithelium. So that is our first epithelial tissue, simple squamous epithelium. Squamous cells, which means they are scale-looking shells, simple because it's one layer thick. Our next epithelial tissue type is going to be simple cuboidal epithelium. Again, simple, we're looking at one layer of cells. It's one layer thick, but now we're talking about cuboidal cells. So remember, these are roughly cube-shaped cells. Again, do not expect to see a perfect Lego brick shaped cell inside of a biological organism. But what you'll notice here is that these cells are roughly as wide as they are long. So they're roughly cube-shaped. They can sometimes be more pyramidal in shape. So especially when we have, say, a tube, a vessel like this, where we have those corners, you might notice that the cell is more pyramidal in shape. But overall, these are going to be roughly as wide as they are long. We're going to have now, whereas the simple squamous epithelium is not going to have projections off of its apical surface, now we're going to get into a few epithelial tissue types that may have projections off of that apical surface. So in our kidney tubules, the apical surface, that surface that touches the lumen, may contain microvilli in the kidney tubules, or it might contain cilia in the bronchioles of the lungs. So we may have projections on our simple cuboidal epithelium that help to either move substances through that lumen space or increase surface area for things like absorption and secretion. Now, already kind of hinted at this, but the functions of our simple cuboidal epithelium are going to be things like absorption and secretion. Here we see an image of kidney simple cuboidal epithelium. Here, these cuboidal cells are doing absorption and secretion work to help to create that urine. We also, though, remember, have this tissue type in areas like the lungs, and if we have cilia on this tissue type, we're actually going to help with the production and movement of mucus through the lumen of that tissue. So places we're going to see this tissue, places we want to do things like absorb and secrete, places we want to do things like move stuff along the lumen. So we're going to find this tissue type in places like the liver, the thyroid, the mammary glands, the salivary glands, the kidney tubules. These are all going to be areas where it's really important for us to absorb and secrete stuff. Then we'll also see it in the bronchioles of the lungs, place where it's really important for us to be able to create mucus to trap any sort of dust particles, pollen, whatever pathogenic substances might be in the air we breathe in. We want to capture that using mucus, and then we want the cilia to help move that mucus along that bronchiole so that it goes up the trachea and we can swallow that away. So here again, we see a microscope image. Here we see the kidneys, and here we see that same drawing of this structure. And what we can see is that, yeah, these cells are roughly cube-shaped in appearance. We can see our basement membrane that is anchoring these cuboidal epithelial cells to some connective tissue. Here in the drawing, you can see that basement membrane highlighted really well. We can see a couple of different lumen here. So we have this large lumen, and then we also have these other lumen there. Remember, the lumen is where the light passes through. So anywhere the light is passing through, we have a lumen. And then lastly, of course, we have the cuboidal cells themselves. And the cuboidal cells, remember when we talk about epithelium, we name the cell in the epithelia based on the shape. So here the cells are roughly cuboidal in shape, so we call it cuboidal epithelium. All right, on to our third tissue type. Keeping with the simple theme, now we have our simple columnar epithelium. So again, simple, remember, means one layer thick. Columnar now, though, we have columnar epithelial cells. So these are our epithelial cells that are going to be really long in appearance, rectangular in appearance. So let me get myself out of the way a little there. And so our simple columnar epithelium, again, is going to be one layer thick, but now we're going to have rectangular or cylindrical cells. So these are going to be long cells. And oftentimes our columnar epithelial cells will have things like microvilli on their apical surface, which are going to help to increase our surface area, to increase our ability to do things like absorption, secretion, filtration. Very importantly, this tissue type may possess these structures called goblet cells. Goblet cells are going to be found in a lot of different epithelial tissue types, and they produce mucus. And we tend to think of mucus as like the stuff that's in our nose, but mucus is going to be any type of substance that's secreted that's going to do things like increase our ability to grab onto things and transport them, create a slick surface for things to pass along. So we also have goblet cells in places like our digestive tract. The functions of our simple columnar epithelium, like I've already hinted at, they possess many of them will possess microvilli. Microvilli, they do absorption. So a lot of our simple columnar epithelial will be in charge of things like absorption. If they have goblet cells, they're going to be in charge of secreting mucus. And in a very special case, we will also have simple columnar epithelium to help with the movements of either ova or embryos within the uterine tubes. So, like their job suggests, we're absorbing, we're secreting mucus, we're moving eggs or embryos. Well, we're going to find this tissue type in places like the gastrointestinal tract, the gallbladder for our absorption and secretion of mucus jobs. We'll also find this tissue type in places like the uterus and the uterine tubes for the movement of the egg and embryo jobs. Here we can see a microscope image of the jejunum. The jejunum is the longest middle portion of the small intestine. Small intestine, we're absorbing molecules from our food. So here we're going to have our nice long columnar cells. What you'll notice is that the apical surface here, because here we see where light's passing through, that must be our lumen. Here we can see that this apical surface is really dense looking at the top. Remember, we talked about this all the way in the first video. H&E stain, hematoxylin and eosin stain, is going to stain proteins pink. Now, if you think about it, our plasma membrane, our cellular membrane, is going to house a lot of proteins. Well, if we have an area of that plasma membrane that we folded over a lot of time to create all of these projections coming off of it, well, that's increasing the amount of proteins we have per square centimeter, or whatever you want to call the space there. So we end up with a really darkly stained apical surface just because we have all those microvilli there that are all going to capture that stain for the proteins that make that plasma membrane. Here you'll notice that it's sometimes called a brush border. Just like a hairbrush, it looks, all of the microvilli look like the bristles on a brush. We call it a brush border because it has that brush-like appearance, and it's the border to the apical surface of the cell. You'll also notice these structures here. We talked about this all the way in section 2 when we were going over terminology. These are the goblet cells, and you'll notice that these goblet cells stain a little bit less darkly. They're creating mucus. Mucus is not a protein-heavy substance, so these are going to stain much less darkly because there's less stuff for that H&E stain to attach to. These are going to be our secretion cells that are producing mucus. You might go, why is it called a goblet cell? Well, if you kind of flip your brain upside down, this looks kind of like a goblet or a chalice that you might see on like a medieval fantasy show. Those really fancy cups they have are called goblets. The goblet cells kind of end up looking like a goblet that's upside down. And then, of course, we have our columnar cells with their nuclei, and then we have that dense basement membrane that is anchoring that basal surface of the epithelial cells to the connective tissue underlying it. All right, now let's go to our funky tissue guy. Remember, we had simple, we had stratified, and then we had that one that was kind of in the middle, pseudo-stratified. Pseudo, remember, is the root word for false, stratified, many-layered. This is the false many-layered epithelium. And what we mean by false many-layered is that all the cells do originate from that basal surface. They are, I should say, are anchored to the basement membrane, but not all of the cells are tall enough to make it all the way to the lumen. So what we end up having is this appearance of many layers. You'll notice these nuclei, it looks like we've got a lot of layers of nuclei here. Looks like we might have a lot of layers of cells, but in reality, we have these basal cells here that are touching the basement membrane, but then don't make it very tall. And then we have these cells here that are attached to the basement membrane, but they go up much further. And so because they go up much further, their nuclei, kind of just because of the pressure of this, the shape and the layout, the distribution of cells on this tissue type, those nuclei end up getting pushed closer to the lumen where there's more space for them. We can appreciate this a little better in the drawing where we have the different plasma membranes drawn out, the different cell membranes indicated. But here you can see that these tissues, all of the cells are smushed. There is some basal surface to each of these cells that's attached to the basement membrane, but not all of these cells are going to have an apical surface that actually touches the lumen. And that's what makes this pseudostratified, false layered. It's one layer of cells in the sense that everybody's anchored to that basement membrane, but it appears many layered because some cells are tall enough to reach the lumen, not everyone is, and that causes the nuclei to kind of end up stacking around each other. So, like I just kind of ran through, pseudostratified columnar epithelium appears multi-layered, however really is only one layer thick because we're considering the fact that everybody is attached to that basement membrane to indicate that this is a single layer of cells. These cells are going to be rectangular or cylindrical in shape because they're columnar. They often are ciliated. Remember, cilia means they've got these long projections off of that apical surface touching the lumen. And oftentimes are going to contain those goblet cells. We already talked about those guys. They're making mucus. So what does this mean? We know there's cilia. That means there's stuff that's getting moved across this tissue type. We know that there are goblet cells oftentimes. That means we're making mucus. We have this tissue in a place of our body where we want to create mucus and move stuff around. That sounds a lot like places like our respiratory tract, where we want to be able to grab any pathogenic substances, any things like spores, pollen, whatever that we're breathing in. We want to be able to grab them and move them out of our respiratory system before they get to our delicate alveoli that are made up of that simple squamous epithelium, which is very delicate because it's very thin. We also are going to find this tissue in portions of the male urethra. Now, if you think about it, the male urethra is going to have to do functions like allow urine to pass, but also is involved in reproductive processes where we're going to want to move things like sperm along that structure. So we find our pseudostratified columnar epithelium in places like the respiratory tract and the reproductive tract of males. Here again, we have our pseudostratified columnar epithelial microscope slide image that comes from the trachea mucosa, so the mucosal layer of the trachea. And we can see that stacked appearance to the nuclei, but in reality, each of these columnar cells is anchored to that basement membrane. We can also see the cilia and the goblet cells. Moving on to a kind of pair of epithelial tissues in the sense that they're both going to be composed of stratified squamous epithelium, but one of these types of epithelia is going to contain live cells at the surface. The other type is going to contain dead cells at the surface. So starting off here with our stratified squamous epithelium, non-keratinized. Non meaning not keratinized. Keratin is a structure that is going to create a really hard external surface. Think of your hair. Your hair is made of highly keratinized cells. So non-keratinized cells are going to be cells that are still soft and alive at the surface of this tissue type. So here we have our stratified squamous epithelium. And what you'll notice is it does look kind of cuboidal, maybe even columnar at the basement membrane, but as we move to the surface, you notice that these cells become increasingly squamous in appearance, increasingly scale-like, smushed, flat. And what you'll notice is that at the surface, and you can appreciate this a lot better in the drawing here, in the stylized rendering, these cells are actually sloughing off at the surface. So where the epithelial tissue actually touches the lumen, you'll notice that these cells flake away. That's a big key in understanding what the job of this epithelial tissue type is. This epithelial tissue is designed for the surface cells to be expendable. What that means is these surface cells can get kind of scraped away, can fall off, And that's fine because that's what this tissue is designed to do. It's designed to resist abrasion and resist penetration by pathogenic organisms. So if you think of something like the inside of your mouth, if you think of the tissue that lines the inside of your cheeks, that tissue is, as we eat, as we talk, as we drink things throughout the day, that tissue is constantly losing cells from the surface just from wear and tear. It also, though, wants to be able to protect against letting pathogens get super deep into the actual bloodstream and whatnot. So by being able to be pulled away and to be able to get scraped off, we're protecting the underlying tissues there. So we're going to find stratified squamous non-keratinized epithelium in places where we want to be able to have wear and tear on that surface tissue. Places like the tongue, the oral mucosa, which is going to be the inside of the cheeks, the esophagus, the anal canal, and the vagina. Now, what all of these areas of the body have in common is that these are going to be tissues that are in areas where the tissues are going to be constantly wet. These are areas that the tissue is not going to dry out. So if you think of places like the inside of your mouth, you're constantly secreting things like saliva that are going to help keep that tissue moist. If you think about things like the anal canal and the vagina, those are also going to be creating things like mucus and other substances that are going to help keep those tissues constantly wet. And because these tissues are constantly kept moist, we are able to have those surface cells remain alive. So you'll notice that all of these surface cells here that touch the lumen are still going to have their nuclei. Those are going to be tissue cells that are still alive. This is going to be different than our next tissue, which is going to be our keratinized stratified squamous epithelium. Keratinized stratified squamous epithelium is going to have a very similar job to the non-keratinized stratified squamous epithelium. However, now we're doing this job in an area of the body where those surface cells are going to dry out. And because those surface cells are going to be drying out, we want to be able to protect the cells below them by creating a waterproof surface. This is where keratinizing the cells and allowing those surface cells to die off comes into play. So stratified, we're many layered. Squamous, our surface cells are going to be scale-like in appearance, very flat. We're going to have increasingly flat cells toward the surface, similar to our stratified squamous epithelium that was non-keratinized. Our basal cells may still be kind of cuboidal or columnar in appearance. However, the cells all the way at the surface will be really flat, scaly, squamous in appearance. However, the big difference here is that as those cells move towards the surface, move towards the lumen, they are going to get increasingly keratinized, which means they are going to be increasingly waterproof, because keratin, if you think about things like your hair, it's a very tough structure, but it is very waterproof. They also are going to become dead towards the surface. The place where we have this tissue type is our skin. If you think about our skin and how often we are losing those cells at the surface of our skin, how often we're scraping away several layers potentially of this type of epithelium, we don't want to lose nuclei all the time. Their DNA is very expensive to make. The proteins in our cells are very expensive to make. We want to maintain as much of those amino acids, as much of those nucleotides as we can within the body. So as these squamous cells develop and mature, we're actually going to start pulling away the DNA from them. We're going to start pulling away those proteins from them, breaking them down, returning them to the live cells, and we're going to allow those cells to die off as they reach the surface, as we are also packing them full of keratin. The keratin helps to prevent water loss. It makes our skin waterproof, which is really good because if you think about bodies, we're essentially bags of water. We want to make sure we keep the water inside. We don't want to let that water get pulled into the really dry atmosphere. So we have this waterproof layer of stratified squamous epithelium that helps to prevent water loss. Similar to the non-keratinized stratified squamous epithelium, big job of this tissue, along with preventing water loss, is going to be resisting abrasion and penetration by pathogenic organisms. So here we have our image of our epidermis, that is the outermost layer of our skin. What we can see is that as these squamous cells are approaching the surface, the lumen, the apical surface there, we are going to get increasingly flat. We're going to start losing those nuclei. You notice that there's far less areas where we have those purple dots, and you'll notice just how tightly packed with protein this outermost layer of cells is. That's from all of that keratin that's built up. We see the most heavily keratinized areas of our epidermis at places like the palms of our hands and the soles of our feet. Makes sense. These are areas of our bodies that are very often exposed to things like abrasion, wear, and tear. Next up, we have stratified cuboidal epithelium. This is, like its name suggests, stratified. That means many layered. Cuboidal, roughly cube-shaped in appearance. This is an epithelial tissue type that is multi-layered with cube-shaped, or sometimes due to like smushing around of other cells, sometimes more round in shape epithelial cells. This is a pretty specialized tissue type. We're going to find this guy in places where we're secreting very specific substances, things like sweat, things like ovarian hormones, and things like sperm. So we're going to find this tissue type in places like the sweat glands, the follicles of our ovaries. Those are going to be the structures within ovaries that are producing eggs, as well as the seminiferous tubules of the testes where we're producing sperm. So this is a very specialized tissue type. It produces just a handful of things, so we find it in very specific places. Here we have a sweat gland, and here you can see that this is many layered because not all of these cuboidal cells are touching that basement membrane, so it is stratified. However, because of the shape of a tube and how tightly packed these cells are, you'll notice that these cuboidal cells are not as perfectly cuboidal looking as our simple cuboidal epithelial cells were. However, most of these cells, yeah, they're roughly as long as they are wide. They're certainly overall more cuboidal in appearance than columnar or squamous. So we're going to call this stratified cuboidal epithelium. And then our very last tissue type we're going to talk about is going to be our transitional epithelium. Transitional epithelium, because this is biology, somebody always has to be special and do something a little bit different from everybody else. This is our one epithelial tissue that's not named after how many layers there are or what the shape of the cells are. And that's because this tissue type does a very specific and special job that requires it to have plasticity in appearance. What I mean by that is this tissue type needs to be able to stretch and contract. And in doing so, we need to have cells that are able to get pulled wide and smushed together. So these cells need to have a plasticity meaning changeability, mutability in shape. Transitional epithelium is called transitional because it transitions from having cells that are really long and drawn out to kind of short and smushed. So the cells themselves transition in shape. Transitional epithelium is multi-layered. The cells at the apical surface, so that surface that is touching the lumen, you'll notice tend to get these kind of umbrella-shaped appearance to them. And we sometimes even refer to these as umbrella cells because they look like umbrellas in that they're kind of rounded at the top. If you have an umbrella, open it up, it's got that nice rounded apical surface. We also sometimes see binucleated cells in this tissue type. So all of our other epithelial tissues had single nuclei, or in the case of keratinized stratified squamous epithelium, might lose their nuclei, have no nuclei. In this case, we occasionally have cells that are binucleated. So bi means two, nuclei. So two cells that contain two nuclei in them. This tissue type, because it has to expand and contract, this tissue type is going to appear thinner when stretched and thicker when contracted. This tissue, if you think of areas where we're going to line cavities, where we want the tissue to be able to distend and return to form over and over and over again, this is going to be a tissue we find in areas where we are producing and storing urine. So if you think about it, your body is continuously creating urine, but it's sometimes creating more urine and sometimes creating less urine, right? So we want places like our ureters, the tubes that carry urine from the kidneys to the bladder, we want them to be able to distend as more fluid goes through them. The bladder, it's going to hold more urine at times. It's going to hold less urine at other times. We want it to be able to distend and then return to form as it holds more urine, empties out, holds more urine, empties out. So transitional epithelium, because it's designed to stretch to allow for that distension of as more fluid passes through it, it helps to protect underlying epithelium from osmotic damage by urine. Urine itself is going to contain things like ammonia, so it is going to be mildly abrasive to underlying tissues. So by having that many layered appearance to the transitional epithelium, we're also protecting underlying epithelial tissues. Like I mentioned, this tissue is predominantly, and in adult organisms, is going to be found in our urinary tracts. There's one other special place where we find it, in the umbilical cord. So during fetal development, the umbilical cord is going to be the structure that carries things like blood to the developing fetus, also carries things like waste products to the placenta. So the umbilical cord is going to have different amounts of fluid passing through it at different times, so it also is going to need to be able to distend and then return to form. So those are our different tissue types. That is the bulk of our new learning for today. In the next video, I'm going to go over how to study and how to prepare for actually coming to lab and what to actually look for in the actual slides that you'll have access to in the lab space. So I look forward to seeing you there for the fourth and final part of this first lab's worth of videos.